Hi, welcome to the nutrition video. So looking at number one, we're going to look at metabolic plasticity and flexibility. This is kind of touched about in the introduction by Museo in his article, but he's going to define metabolic plasticity or flexibility as where mitochondria are able to switch between substances, are fats and sugar. So I just want to highlight, I'm going to write metflex, but the battle between sugar and fats. So sugar and fats. It's important to look at how these mitochondria are responding to modern day diet and the pressure they have to choose between fat as a fuel source when glucose reserves are threatened. And additionally, the increased supply of oxidation of fatty acids leads to our cellular accumulation of acetylcholine, NADH, and ATP, which inhibit the pyruvate dehydrogenase. So looking at number two, our CPT1 is the gateway for fatty acids, enter the mitochondria, and acetylcholine is the gatekeeper. So when drawing this out, we're going to have AMPK. So this is going to regulate our fatty acid synthesis and our B oxidation. So it is a negative regulator of our acetylcholine carboxylase activity, and our ACCs can catalyze the conversion of acetylcholine and methylcholine. So I'll put methylcholine there. This is also going to inhibit our carnitine. I don't know if I can pronounce this, but it is our CPT1. So this is going to catalyze the transfer of the fatty acetyl group from the acetylcholine. So we'll put our CPT1 here with the relationship of acetylcholine. And we're going to have our B oxidation. This is going to happen in the mitochondria. So put the oxidation here. And we also have fatty acid synthesis. So we're going to be preparing it for our transport from the cytosol into a mitochondria. And as I said, it's going to be used here for our B oxidation. So looking at number three, we are going to be looking at the metabolic congestion and metabolic road rage. So it's important to look at our metabolic congestion as the battle between substrates within our mitochondria. So we are going to be fighting between these substrates, and it is something that has not always been evident, especially in our hunter-gatherer days, but we're in this stage of chronic overfeeding. Competition between these substrates is going to happen. We're going to have less cooperation between mitochondria, and we're going to have more metabolic inflexibility. So it also is important to just look at our amino acid supply, and emerging evidence is going to suggest that our increased amino acid supply and catabolism might also contribute to obesity-related perturbations in our fuel use. So when looking at the road rage, this is going to be where we're going to have a situation where it's going to be distorted, and we're going to have conflicting signals between the products of our CPT1, PBK, and BCKD. As these tensions continue to escalate, our three substrates are going to battle for which one is the right of way. And the mitochondria are confronted with an ever increasing decision of they're going to have this nutrient burden. They're going to have to choose which one they want to go with. So looking at the next question, number four, OXPHOS is a demand-driven process regulated by ADP availability. Explain why this is so vital to understand. So I guess one mechanism that is important in OXPHOS is controlled by this is our respiratory control. So we're going to have isolated mitochondria here, and our OXPHOS activity is going to be eliminated by the availability of substrates for our ATP synthesis, ADP, and phosphate. Our OXPHOS activity is going to increase when our ATP utilization generates ADP and phosphate. Our mitochondrial OXPHOS is also going to generate ATP by utilizing our electrochemical gradient created by our mitochondrial electron transport. This pathway is profoundly important in on how it affects the energy metabolism and calcium homeostasis. So when looking at OXPHOS in general, the main important thing to happen here is we're going to have, and it's going to happen in the intramembrane space. We're going to have NADH go to NAD. I'm just going to abbreviate it for shorter since I've had a little bit of a hard time trying these out. But then we're going to have FADH2. This is going to go to FAD, sorry. And then we're going to have O2 going through our coenzyme C and our coenzyme Q. our coenzyme Q is going to help facilitate the electrons to the correct spot, and we're going to have this moving into H2O. And then we're going to have our ATP synthase. This is going to go to ATP. So we'll put ATPS, and this is going to create about 36 to 39 ATP. Just an important thing to look at when we are highlighting our OSPOS. So next question, discuss why greater electron availability in the mitochondria does not always lead to greater ATP production, and describe what happens to this excess electron when there is back pressure in the protein chambers. So greater availability of electrons does not always lead to greater ATP production because our electrons are in the process of continually being used up. This is going to occur only in a transfer of electrons, and they're going to pass along a chain from protein coupled complex to the next protein coupled complex until they're donated oxygen. And this electron donor is going to act as our limiting factor in the situation. So if there is an excess electron, this can cause formation of superoxides, which can end up harming the cell. And these terminal oxides in the thylakoid membrane play an important role in addition to any function in respiration. So namely the transfer of excess electrons generated from PS2 to oxygen. And this is going to be important in preventing the overreduction of superoxides in our system. So next question, explain exercise snacking, how it relates to carbon overload in the mitochondria. How can this technique be combined with nutritional changes to mediate congestion in the ETC? So interesting to look at. Exercise snacking regimens are where individuals are going to exercise vigorously 5 minutes for a short period before consuming a standard meal size. So these routines were found to improve glucose homeostasis more than isoenergetic regimens comprised of traditional feeding and also exercise patterns. So it's kind of interesting, I'd say, that our glucose homeostasis would be better than a meal just for exercise for a short time, but it's just very interesting to note that. And likewise, the interruption of prolonged sedentary behavior with repeated bouts of low-grade activity of daily living has been shown to also improve our postprandial glucose handling in people with diabetes mellitus 2. These observations are also consistent with the premise that periodic episodes of accelerated carbon combustion were evident in our hunter-gatherer ancestors. So I would say if I was recommending this to somebody, I would probably try to get some cardio in, but I also think it could be a very valuable lesson to someone kind of wondering when to eat and to work out in conjunction with one another, that going for a short run or playing some basketball, and then if you're going to eat a larger meal, do that afterwards. But it's just, I'd say it's something that's important and shows great research with our metabolic flexibility in terms of the glucose that we were having in our body. And lastly, discuss specific ideas and considerations of how the nutrition field could trade in eat less, move more. So when looking at this, I found a study from Carlson. This was one of the studies that was cited towards the end of the article, but they took healthy human volunteers and they did 60 hours of fasting, collected blood samples at 12 and 60 hours, and they observed a decrease in plasma glucose by 30% and insulin by 50%. This was a significant increase in the extent of the lipolysis and fat oxidation, and they also had an increase in the extent of their protein oxidation. Overall, they were able to show improvement in their lipid profile after intermittent fasting. I think it's just an important thing to talk about is eating less is important, but I think it's just more important what you're putting in your body. I feel like for me, over my journey, it's because I feel like when you're growing up and you're going through that puberty stage, it's you got to eat what you want, but you just eat as much as possible. It doesn't matter what you're putting in your body. But I feel like as I've gone into college, it's not how much I'm eating, it's what I'm eating. So if I eat this large meal of healthy food, I mean, I feel pretty similar to if I'm eating a smaller meal of healthy food. So I would recommend for anyone just to have this daily inconsistent exercise, and then you don't always need to worry about as much about what you're putting in your body. So I think it's very, very important what you're putting in your body, but you can have some of the normal things if you don't want to get it out of your diet. Like if I want to have some ice cream, but I'm still working out on a consistent basis, this is going to help my metabolic flexibility in an important way. So looking at our biochemistry, the study that was also presented also confirmed that they also had an expression of muscular expression of genes, like their GSK3. So this is responsible for the regulation of glycogen synthesis pathways, and it's going to favor our glycogen reposition. So we're going to actually have gene expression and changes in um, our molecular DNA just based on following fasting and regimens that are going to improve our metabolic flexibility. So I think that's just so important. And our CPT1 is also important. So we can also have increased expression of that in our muscle tissue and in fat oxidation when we're eating less and working out more. But that's about it for my midterm video. I think I touched on everything to some extent pretty well. I hope this wasn't, it might be a little long, but thanks for listening and have a great day.